Hello friends, welcome back to my channel BioFric. In today's video, I'll be talking about phosphotransferase system, wherein a phosphate group from high energy molecule phosphoenol pyruvate is transferred onto the series of enzymes in this system, and finally onto the substrate like glucose or mannitol or mannose, which is the energy source for prokaryotes. Now, in order to understand this system, first let's look at this diagram, wherein this is the cell wall and there is this integral transporter protein through which energy source or the substrate enters the cell. This is the internal of the cell, and these all enzymes are intracellular enzymes. So phosphoenol pyruvate, as we all know, it carries a phosphate group. Now that phosphate group is transferred onto E1. This is an intracellular enzyme, and when it transfers its phosphate group onto E1, the pyruvate is released, and this phosphate group from E1 is transferred onto the next enzyme in the series, that is histidine protein. And this phosphate is later transferred onto E2A and then E2B, and finally E2B transfers that phosphate group onto the glucose that has entered the cell. Now, in this whole scenario, cell prefers the simplest source of energy. If glucose and lactose is there in the environment, the cell usually prefers the glucose, that is simpler form of sugar, right? So similarly, to understand this in a better way, let's look at the three different situations. Now, first one is catabolite repression. In catabolite repression, your usual glucose is in lesser quantities in its surrounding, and this is your E. coli cell, and this is your normal cycle. This is your normal series of transfer of phosphate group. Now, what happens is, when glucose is not there in the environment, it cannot simply enter the cell. E2A retains the phosphate group. Now, it's in phosphorylated situation. It's phosphorylated, and phosphorylated E2A cannot go and bind to adenylate cyclase. Now, this adenylate cyclase is actually the enzyme that converts ATP to cyclic AMP. Now, phosphorylated E2A cannot go and bind to adenylate cyclase, so ATP is converted to cyclic AMP, and the cyclic AMP goes and binds to some operon that will express itself, and it will allow the entry of non-preferred substrate like fructose 6-phosphate. So this whole thing occurs inside the cell. This is the concentration that increases inside the cell. Now, in this situation, we saw that there was starvation going on, but in the next condition, glucose is there is enough of glucose, and as well as there is fructose, but then cell wants to take in just glucose and not fructose. So how does it do that? It is very simple. E2A will phosphorylate glucose to glucose 6-phosphate, right? It will transfer that phosphate group onto the glucose. Now, dephosphorylated E2A inhibits the adenylate cyclase. It will inhibit adenylate cyclase, and thus it cannot convert ATP to cyclic AMP, which won't go and bind to operon, and hence this transporter will remain in the closed state. It won't accept the entry of fructose inside the cell. But now, in this situation, what is happening is inducer expulsion. Now, there was starvation state going on previously, and the cell had adapted to fructose, and it was taking in the fructose, so which was converted to fructose 6-phosphate. But now suddenly the cell senses that there is glucose in the environment back, which is the simplest form of sugar. How will it go back to this normal state? So it's done by this HPR, which is histidine rich protein, that will steal the phosphate group from fructose 6-phosphate. Now, fructose 6-phosphate is again converted to fructose. It will be sent back via the transporter, and now we know that the phosphate will be transferred onto E2A. E2A will transfer that phosphate onto E2B. And that phosphate will be transferred into the incoming glucose molecule to glucose 6-phosphate. So glucose 6-phosphate will be formed. Now, what is happening in this whole procedure? E2A is again dephosphorylated. Dephosphorylated E2A can go and bind and inhibit the adenylate cyclase. Thus, there will be no formation of cyclic AMP, and it will shut this pores again, and there won't be any entry of fructose. So that was about the phosphotransferase system.